Start that one around, please. Okay, it looks like it's about time to get started. I just checked my audio, so it looks like I have audio. Can everybody, pardon me. Can everybody joining by Zoom just confirm that you have audio and video for me with a thumbs up or in chat? Good, thank you very much. All right. Let me just get the chat up here. Good. Okay. So first, you're probably wondering about your tests. I have a couple more makeups to give. Some people had excuses. So I'll make the feedback, your answers and everything available as soon as they complete their exams. Secondly, quizzes. So I'm going to give you guys today off from quizzes because we just had a test on Monday. But we'll start up again on Friday. And Friday's quiz will be on what we cover today and what we cover Friday. Okay. And again, let me just emphasize to you guys, quizzes are actually a very good thing for you guys. And we really should try to fit as many as we can into this semester, because I only count your 20 highest quiz grades. So the more quizzes we have, the more of your lowest dropped grades there will be. And also the more study material you have, the more questions are going to be on the exam from the quizzes. Okay, so we'll get started up again with quizzes on Friday. And unless there's some questions, I think that's about it. So let's get back to chapter 11. So we had just started on chapter 11, just got the first couple slides out of the way. Chapter 11 has to do with mitosis. Okay, mitosis is the way that eukaryotic cells divide. All right, in multicellular organisms like us, this is the way that we replace older dead cells. This is the way we grow, and this is the way we repair damage as well. In prokaryotic, not prokaryotic, but in single-celled eukaryotes, remember this is only for eukaryotes. In single-cell eukaryotes, this is the way that they reproduce. And there is this organized series of events when a cell reproduces called the cell cycle. And we were just able to go over this and a little bit more before we ran out of time on Friday. So we can divide, just to review quickly, we can divide up the cell cycle into two major divisions. We have mitosis, or M phase, which is right here, this yellow arrow from here to here. And then we have interphase. Okay, interphase is everything here back around to mitosis again. Okay, so that's interphase. Okay, interphase can be broken down further into three parts, G1, S, and G2. Okay, G1, or gap 1, is an interval of normal cell growth and function before DNA replication. So in other words, during G1, your bone cells are doing what bone cells are supposed to do. Okay, during G1, your liver cells, your hepatic cells are doing what liver cells are supposed to do. Just normal growth and function. Once a cell crosses over from G1 into S phase, so this is S phase here. S phase stands for synthesis. This is where DNA is replicated. So if we're using human cells as an example, human cells normally have 46 chromosomes. Okay, we get 23 chromosomes from our mother, 23 chromosomes from our father, for a total of 46 chromosomes. In S phase, we need to make an extra set of chromosomes because if we're going to divide the cell in two, each new daughter cell is going to need a full set of chromosomes. Okay, so after S phase, if we're talking human cells, instead of having 46 chromosomes, we have 92 chromosomes now. Okay, because we have an extra set. G2 or gap 2, this is a period of growth in preparation for division. So in other words, during G2, a cell is going to make sure that it has enough stuff to divide into two cells. It's going to be, there's checkpoints. You know, does the cell have enough mitochondria? Does the cell have enough endoplasmic reticulum? Does it have enough plasma membrane to divide in two? And during G2, it'll grow in order to make sure it has enough stuff, basically, to divide in two. Okay? Then we have the different parts of mitosis. So there's prophase, metaphase, anaphase, telophase. We'll talk about these in more detail later. And one thing missing from this cell cycle, between mitosis and G1, we have cytokinesis. So cytokinesis is where the parent cell physically divides into two new daughters. That's a horrible daughter cell, but you guys get the idea. Okay, and this is part, so between telophase of mitosis and G1, we have cytokinesis. All right, looks like I'm getting better at drawing with the mouse. Still not good, but better. That's not bad at all. Cytokinesis. All right. So those are the parts of the cell cycle. And again, you guys already had the lab for this, so this should be somewhat familiar to you. Okay, and this is just, I already went over this in the last slide. So interphase, we can divide into three parts, G1, S, G2. Mitosis, 4 parts, prophase, metaphase, anaphase, telophase. And right here, again, we have cytokinesis, or the physical division of the parent cell into two new daughter cells. Okay, so chromosome, this is about where we left off on Friday. So a chromosome is a single molecule of DNA inside of a cell. Like I said before, humans normally, so before the cell enters S phase, human cells have 46 chromosomes. Okay, again, you get one set of 23 from your mother, one set of 23 from your father. So this is an example. I talked about this briefly on Friday before we ran out of time. So this is what's called a karyotype. A karyotype, so these are the actual pictures of actual chromosomes from a cell. And you can see we have 23 pairs of chromosomes for a total of 46 chromosomes. Okay, chromosomes vary in size, so you can see there's some big ones and some smaller ones. Contained on these chromosomes are genes. Okay, each gene codes for a protein. So we learned earlier in the semester that the amino acid sequence of a protein is encoded in DNA. So each of the genes contained on these chromosomes contains the amino acid sequence for a protein. Each chromosome replicates during S phase, we talked about that again, into two identical sister chromatids. And the two chromatids are held together by a centromere. All right, it's going to help if I draw this on the board. So let me stop the share for a second. And let's go back to the camera. Okay, I'm just going to draw over here instead of, okay, I need something to write with. Okay, so you might recall when we talked about DNA. So DNA is an extremely long molecule. Well, I'll show you an example of that a little bit later. These are very, very, very long molecules. A single chromosome, an average single human chromosome, is actually about 2 inches long if we were able to stretch it out just to as a string of nucleotides. It'd be about 2 inches long. Okay? And you got to remember that that fits inside the nucleus of a cell that's only about it's about 6 micrometers, or about 6 millionths of a meter in diameter. So in order to fit that inside the cell and organize it, we have to wrap it up. Okay, you might recall from an earlier class that there are proteins associated with DNA called histones. Okay, and histones are basically like spools that DNA wraps around. So inside the cell, these big DNA molecules are kind of wrapped up and condensed down. All right. Also, after S phase, so here's one that's kind of wrapped up and condensed down. Here's another. Okay, so when a cell gets to mitosis, when it gets to around metaphase of mitosis, we'll talk about that later. The chromosomes look like this. They look like kind of like the letter X. Okay. Each side of the letter X, so if we can draw like a dotted line down the middle, each side of the letter X has one sister chromatid. These are sister chromatids. And what the sister chromatids are is they're exact copies of the same chromosome. So let's just say, so remember, like I said before, so we have 46 chromosomes, one set of 23 from your mother, one set of 23 from your father, if we're using humans as an example, okay? If we say, let's say this is chromosome number 1 that you inherited from your mother. So one half of this, one sister chromatid, is chromosome number 1 from your mom. The other sister chromatid is an exact copy of chromosome number 1 from your mom, okay? And these are connected at a place called the centromere. So this place where it kind of makes an X, this is the centromere. All right. So sister chromatids. After we go through S phase, we actually have 92 chromosomes rather than 46. And we have 2 identical sister chromatids that are connected at the centromere. Okay. And just let me, is this monitor work? Good. I put in a ticket for that the other day. All right, let's go back to the PowerPoint. So if we look a little more closely at this karyotype of human chromosomes, it's kind of hard to see, but if you look closely, you can see the 2 sister chromatids and the centromere. Where's a good one? The 2 sister chromatids are pretty close together, so it's kind of hard to distinguish them as separate. If we look at chromosome number 16 down here, I think it shows it best. So you can see one sister chromatid on this side, another sister chromatid here, and then this kind of constricted area. This is the centromere. Chromosome 10 looks pretty good too. So you can see one sister chromatid here, another sister chromatid here, and then you can really very clearly see the centromere right about there. Okay, between the end of S phase and the end of mitosis, a human somatic cell has 46 times 2 or 92 chromosomes. I've talked about that before. There's a term here that you might not be familiar with, so let me just define that term before we move on. And that's this word somatic. What somatic refers to are the cells of your body other than gametes, other than reproductive cells. Reproductive cells are going to differ in the number of chromosomes for obvious reasons. So, and we'll talk about that during the next chapter, development. So somatic refers to cells other than reproductive cells in your body. Okay, so between the end of S phase and the end of mitosis, the human cells other than the reproductive cells have 92 chromosomes. That's 2 sets of 46. Upon completion of mitosis, each daughter cell will receive 46 chromosomes. Okay, so during mitosis, this is where we're physically going to divide the DNA into 2 new nuclei. Then after cytokinesis, this is where we're going to physically divide the parent cell into 2 new daughter cells, and we'll end up with 2 new daughter cells, each with a complete set of chromosomes. Okay, so here's another karyotype. And on Friday, I asked if anybody could tell me that other karyotype we saw. I asked if anybody could tell me if that karyotype came from a male or a female. Looking at this karyotype, does this karyotype come from a male or a female? Anybody in class or on Zoom? Can anybody tell? Female, somebody from Zoom, Stephanie from Zoom says female. Female's correct. We have 2 X chromosomes here and here. So if you're XX, so you want to look at the sex chromosomes if you want to figure out if it's a male or female. That's what decides the sex. If we have 2 X chromosomes, that's a female. If we have XY, it's a male. You guys haven't seen a Y chromosome yet, but a Y chromosome is much, much smaller than an X chromosome. So this is obviously a female. Okay, XX. And again, we can see the 23 pairs of chromosomes in this karyotype. Okay, next I'm going to go through the stages of mitosis. So remember that there are 4 stages of mitosis. And again, you guys had a little practice with this in lab because the labs and the lectures are a little out of sync right now. So there's 4 stages to mitosis. We have prophase, metaphase, anaphase, and telophase. And again, specific things happen at each phase of mitosis. Prophase, this is where the most is going on. So there's about 6 things you're going to have to remember that happen in prophase. The first thing that happens in prophase is the chromosomes condense. So, and again, in last week's lab, no, last week you guys had the test. The week before you had the mitosis lab, right? So you might notice that when you looked at cells that were in interphase, the nucleus was a pretty even, a pretty homogeneous blue staining color, except you might see that one dark spot called the nucleolus. That's because the DNA was uncondensed. Remember, the DNA, these are very large molecules. So when they're not wrapped up in histones and condensed down, they're very large. One of the first things we have to do in order to divide this DNA into 2 new nuclei is to wrap up and organize all that DNA. So when we wrap up and organize this DNA, it condenses down into these little X shapes with 2 sister chromatids. So that's the first thing that's going to happen. So we're going to see the chromosomes start to condense down into sister chromatids. Number two, new microtubules are made. Microtubules, you might recall, are a fiber of the cytoskeleton, which is the network of protein fibers that give cells shape and structural support. Okay. Microtubules also play a role in cell division. So they make up something called the mitotic spindle. The mitotic spindle is what's actually going to separate out these sister chromatids into two new sets of chromosomes. Okay, so that mitotic spindle is important. So we need microtubules in order to assemble that mitotic spindle. Number three, microtubules begin to assemble around two pairs of centrioles into a mitotic spindle. Okay, on this diagram, the two centrioles are here. Okay. And what the centrioles are, I should probably write this down. Let me stop the share and go back to the board quickly. What centrioles are, they're a type of modified microtubule. So they're proteins that are very similar to microtubules, but they help to organize the mitotic spindle. Eraser. So centrioles. So these are a special kind of microtubule, spelled that wrong. That organizes the mitotic spindle. Okay, so centrioles. So let me just talk to the Zoomers real quick. So on my screen, everything is backwards. Is it the same for everybody doing Zoom? That's really weird. Looks okay to, okay, people are saying looks okay. No, for some reason on my screen. Okay. Well, as long as you guys can see it okay, that's fine. Okay, so centrioles are a special type of microtubule that organizes the mitotic spindle. Let's go back to the PowerPoint. Okay, so first three things going on. Chromosomes condense, so we need to get these chromosomes organized in order for to get two new nuclei. New microtubules are made, and these new microtubules begin to assemble around two pairs of centrioles into a mitotic spindle. Again, the two pairs of centrioles are these two things in red, right? Whoops, where's my drawing tool? These two things in red right here. Okay, next, the two pairs of centrioles begin to migrate to opposite sides of the cell. Pardon me a second. Okay, so what's going to happen is, so right now we have two centrioles on the same side of the cell. These are going to start to move apart. So eventually we have one pair of centrioles on one side of the cell and another set of centrioles on the opposite side of the cell. Okay. Next, number 5, the nuclear envelope breaks down. So looking at this, these are all parts of the nuclear envelope. So Of course, remember the nucleus contains and protects the genetic material, but if we're going to break it up into two sets, one for each new daughter cell, that nuclear envelope's just in the way, okay? So we're gonna have to temporarily get rid of that. And later on, it will reform two new nuclear envelopes around the two new nuclei. Okay, so nuclear envelope's in the way, we just need to get rid of that for a little bit. Number 6, the microtubules of the mitotic spindle begin attaching to the centromeres. Okay, so now remember, the centromeres are that little part of the X right here where the two sister chromatids attach. So the microtubules are actually going to attach to the sister chromatids at the centriole. Okay, so that's prophase. So again, in prophase, number 1, so the chromosomes are going to begin to condense. We're going to make new microtubules. Microtubules are going to start assembling around the centrioles. The centrioles are going to migrate to opposite poles of the cell. Nuclear envelope breaks down. Microtubules start attaching to the sister chromatids at the centromeres. Okay, so this is a picture of DNA. This is actually from a bacterium, not from a human cell. But looking at this, you can get the idea. So DNA, like I said, is a huge, huge, big, long molecule. Okay, even a single human chromosome is about 2 inches long. And we have 46 of those inside of the nuclei of our cells, which are, again, the nuclei are only about 6 micrometers or so in diameter. So you can imagine, without organizing this, if we tried to split this into 2 separate sets of chromosomes, it would get all tangled up. The DNA would probably break. It would just be impossible to do. So it's very important that we organize and condense this down. Okay. I don't know if I can remember the exact math on this, but if you take all of the chromosomes in a single cell, so remember each chromosome is about 2 inches long, and stretch it out so it's a single chain of nucleotides, then put all of those 46 together, that comes to about 2 meters or about 6 feet. Okay, that's for each cell. Now, when you consider that we have about 1 trillion cells in our body, if you line up all of the DNA, if you can stretch out all of the chromosomes into a single chain of nucleotides, put them all end to end, then actually that will be long enough to go from, how does it go? To go from here to the sun and back several times. Okay, so that's a lot of DNA. So again, this just is to emphasize how big this molecule is and how important it is to condense it down in order to organize it so that we can get one set of chromosomes to each of the 2 new daughter cells. Okay, so I think we talked about histones before. So this is a histone. And like I said before, these are proteins that wrap and organize DNA. So we can see, so here's a histone, and we can see the DNA wraps around the histone much in the same way that thread wraps around a spool. Okay, so this is going to help to organize it. There's actually several more layers of organization that will condense this DNA down even more until eventually we see those X-like structures with the 2 sister chromatids together. Okay. So some terminology here. When DNA is associated with proteins, we refer to that as chromatin. So chromatin, that's CHROM A-T-I-N. This is DNA associated with proteins. So this is DNA plus, there we go, plus histones. Oh, it's a horrible E. All right, you guys get the idea. Okay, and when we have one histone associated with DNA, we refer to that as one nucleosome. So this is one nucleosome. Looking at C over here, this is an actual picture. This is an electron micrograph of DNA. And we can see the individual nucleosomes. So right here is DNA associated with histones. So these are all nucleosomes here. At this stage of organization, we call this the beads on a string level of organization, because that's basically what it looks like. Okay. But again, there's several levels of organization above this that's going to condense down the DNA even more. That goes beyond the scope of this class. If you guys take genetics in the next year or so, you'll talk a lot more about those other levels of organization that we see. Okay, so here's some pictures. You guys probably saw some similar pictures in lab, and probably looked at some slides at the different stages of mitosis as well. So remember that a couple things happen during prophase. Let me just clean this up a little more. Whoops, not what I wanted. Okay, so here we're looking at the cell at interphase. So remember, interphase, all of this DNA is going to be uncondensed. So it's just going to be kind of evenly spread out throughout the nucleus. So we see this very even, homogeneous, blue staining nucleus. Okay. When we enter early prophase, remember one of the first things that's going to happen is the DNA is going to start to condense down into sister chromatids. So you can see how, comparing here to here, you can see how the look of the nucleus has changed. So instead of an even blue color, we have these, we can see the discrete chromosomes starting to form. We see a more granular sort of look to the nucleus. Okay. Late prophase, by late prophase, all of the chromosomes are condensed down. So here we can see the distinct sister chromatids. So we can see there's molecules, each molecule is condensed down. Also notice over here, we can kind of see the nuclear envelope. We can see the nuclear envelope is completely broken down here as well. Okay, that's prophase. So there's actually the most to remember about prophase. There isn't quite as much to remember about metaphase, anaphase, and telophase. So next, let's talk about what happens in metaphase. So in metaphase, the two centriole pairs arrive at opposite sides of the cell, forming a spindle axis. So notice, so here's one pair of centrioles. The other pair of centrioles has arrived at the opposite pole of the cell. And now we have a completely assembled mitotic spindle. So again, these pairs of centrioles are going to organize these microtubules into the mitotic spindle. Next, the microtubules of the mitotic spindle pull the chromosomes until they are aligned along the middle of the cell, forming the spindle equator. Okay. So right here is the spindle equator. I'm sure you guys, when you were doing the mitosis lab last week, you noticed that mitosis was one of the easiest phases, metaphase was one of the easiest phases of mitosis to pick out, because all the chromosomes are lined up along the spindle equator. And it's really easy to pick out under a scope. Okay? So that's about it for metaphase. So again, not much to know about these metaphase, anaphase, and telophase. So the centrioles arrive at opposite poles of the cell, and this forms the spindle axis, and the chromosomes are pushed and pulled until they're lined up across the spindle equator. The next phase of mitosis is anaphase. Okay? And this is the phase of the 4 phases of mitosis. This is the one that happens the fastest. It's going to change between cells, but about 45 seconds to a minute is all that it takes for a cell to go through anaphase. And you guys probably noticed when you were counting cells in lab 2 weeks ago that you probably didn't see a whole lot of cells that were in anaphase. So in anaphase, the 2 identical sister chromatids previously joined at the centromere move apart. Okay, so these microtubules that are attached at the centromere start to contract, and they pull the sister chromatids apart so they're no longer attached to the centromere. Okay, so this is going to give us, one set of chromosomes on one pole of the cell, one set of chromosomes on the other, and each of the 2 new daughter cells are going to receive one new set of chromosomes. Okay. Some microtubules of the mitotic spindle attached to the centromeres pull the chromatids to opposite poles of the cell. So those would be these microtubules in red. Other microtubules push the centrioles apart. Okay, so they're not shown here, but I'm going to draw in another microtubule here and another microtubule here. Okay. So actually where these 2 microtubules overlap, there are motor proteins. So here's a motor protein, here's a motor protein, here's a motor protein. And these motor proteins are going to attach to the microtubules and push them apart. So this microtubule is pushed this way, this microtubule is pushed that way. Okay? And notice how the cell's elongated. So these microtubules that push apart are going to push the centrioles farther apart, and that's why we get this oval shape. So it's going to elongate the cell and make it longer. Okay, so that's about it for anaphase. So remember, this happens very quickly in the cell. Some microtubules are going to contract. That's going to pull apart the sister chromatids so they're not attached at the centromere anymore. Some microtubules are going to push. That's going to elongate the cell and push the centrioles farther apart. Okay, the last phase of mitosis is telophase. In telophase, the sister chromatids are going to move even farther apart. 2 nuclei are going to begin to reform in opposite sides of the cell. The mitotic spindle disassembles. So once we get the chromosomes where they need to be, we don't need the mitotic spindle anymore. So the mitotic spindle is going to disassemble. And finally, the chromosomes are going to decondense. So instead of seeing these sister chromatids that are condensed and easy to visualize, they're going to decondense. And finally, 2 new nuclear envelopes are going to form around the 2 new nuclei. All right. So again, the sister chromatids move farther apart. 2 new nuclei begin to reform. So the nuclear envelopes are going to start to reform around the 2 new sets of chromosomes. We don't need the mitotic spindle anymore. That's going to disassemble. The chromosomes don't need to be condensed anymore, so they're going to decondense. A good way to think about that is if you're going on vacation and moving or moving, let's just say you're going on vacation. So you're basically, you take your stuff and you pack it into your suitcase. Or if you're moving, you take your stuff and you pack it up into boxes. Okay, that's basically condensing your stuff down the same way that a cell condenses down its DNA, right? When you get to your new house, what do? You unpack your stuff out of the boxes. Okay, that's what's happening here in telophase, where the chromosomes are decondensing. Okay, so you get to your new house, basically you're uncondensing your stuff out of the boxes. When the new nuclei start to form, we can decondense the DNA and unwrap it and decondense it. Okay, so let's look at some more pictures. So here we have late prophase going into metaphase. So we can see that the two centrioles here and here are moving farther apart, not quite at opposite poles of the cell yet. We can see that the microtubules of the mitotic spindle are attached to the sister chromatids at the centromere, here, here, and here. All right, and as these two centriole pairs move to opposite poles of the cell, they're going to kind of push and pull the sister chromatids until they line up across the spindle equator. So here's metaphase. Metaphase, the two centrioles have arrived at opposite poles of the cell, and we have all of the chromosomes lined up along the spindle equator. Okay, anaphase. So some microtubules are going to contract. That's going to pull apart the sister chromatids that were previously attached at the centromere. Some of these are going to pull them apart. Some of these microtubules of the mitotic spindle, we can see here, are going to push against each other. That's going to move the centrioles even farther apart and elongate the cell like this. All right. After anaphase, we're into telophase. We can see the two new nuclei starting to form. So we can see a new nuclear envelope forming around each of the two new nuclei. We can decondense the chromosomes. So you can see, again, we're getting an even blue color because of the decondensed chromosomes. Okay. Finally, after this, we get cytokinesis. Cytokinesis is the physical division of the cell into two new daughter cells. And then after cytokinesis, the cells are going to enter the cell cycle at G1 of interphase again. Okay, so cytokinesis. So I told you at the very beginning of the lecture, between telophase of mitosis and G1 of interphase, we have cytokinesis, or the physical division of cells into two new daughter cells. This happens differently in plant cells and in animal cells. And again, you probably had a chance to observe this in lab last week. So in plants, vesicles along the spindle equator fuse to form a cell plate. Okay, we can see that here. Whoops. I'm sorry. Okay, so again, we can see that here, vesicles forming a cell plate. Eventually, cellulose is deposited in the cell plate, forming a cell wall, okay, like this here. Plants, it's a little bit different. So in animals, it's a little bit different than plants. So in animals, the dividing cell pinches in half along an indentation in the plasma membrane called the cleavage furrow. Components of the cytoskeleton pull the two daughter cells apart. So if you can imagine, right here where all these arrows are, this is the cleavage furrow. In the cleavage furrow, there's a contractile ring of cytoskeletal filaments. Okay, and that ring is just going to get smaller smaller, smaller, smaller as it contracts until eventually it pinches off the animal cell into 2 new daughter cells. Okay, so 2 different ways we have a we have a cell plate that forms in plant cells, and we have a cleavage furrow that contracts and pinches off the cell in animal cells. Okay, we're out of time, and it's really too bad that I can't show you. I don't have time to show you these mitosis videos. I'll post them on eCampus. Friday when we come back from class, that will be the first thing that I show you. So again, let me just remind you, since we're out of time, quizzes will start up again on Friday. After I get all of the makeup tests done, you'll be able to look at your questions and answers from the test on Monday. Okay, if there's no questions, I'm going to stop the Zoom meeting here, and I will see you guys either on Zoom or in class on Friday.